and welcome. My name is Sandra Evans and I'm the Global Sales Leader for the Solus Product Portfolio, which is now part of PerkinElmer's Food Safety Solutions. I'm delighted to be able to give a brief overview of Solus and our capabilities and want to take this opportunity ahead of the Meat and Poultry Symposium on the 6th of October to highlight our newest addition to our product portfolio, Solus Listeria monocytogenes, which gained AOAC PTM certification in August of this year. Solus Scientific develop and manufacture pathogen detection kits exclusively for the food testing market and provide a highly efficient method for the detection of Salmonella, Listeria and E. coli 157 in a wide range of food matrices and environmental samples. Each system comprises of a specifically designed immunoassay kit, dedicated selective enrichment media, and an automated liquid handling system. Our pathogen testing systems are robust, reliable and validated by external laboratories to ISO 16140 certification and certified by AOAC Performance Testing Method and the OMA program. Solus Scientific started as an immunoassay development company in 2009 and merged with a distribution company in 2011. We launched Solus Salmonella and Listeria in 2013 with ISO 16140 certification and followed by E. coli 157 in 2015. And in the UK and Ireland, this led to us becoming a leading supplier of pathogen detection kits in this territory. From the successful introduction of Solus assays, we then launched our products internationally and quickly gained a global footprint in the food testing market. We achieved AOAC PTM for Solus Salmonella and Listeria and launched our products into the US market into 2016 and subsequently gained AOAC and ISO certification for Solus One range, our next generation immunoassays, the next data result capabilities. In 2019 we were acquired by PerkinElmer and as part of the acquisition enabled us to add AOAC extension to scope for Solus One Salmonella for herbs, spices and confectionery products. And in 2020 we have continued to follow our development programme and at the beginning of the year we gained AOAC OMA certification for Solus One Salmonella. Then in August this year we launched Solus Listeria monocytogenes with AOAC PTM for food and environmental samples. So when we talk about our system, we talk about the kit, the automation and the dedicated media. But we need to remember that the Solus kits can also be run manually with a washer reader. So why do we develop dedicated media with our kits? We believe that testing is only as good as the enrichment. Unlike most other method developers, Solus have developed enhanced traditional enrichments alongside our assay kits to improve recovery, growth and therefore the detection of the pathogens, especially in difficult samples. This ensures the media is consistent in quality and performance, so gives reproducible results in the long term. We work closely with our suppliers to ensure dependable sourcing of the raw materials to safeguard this reliable quality. Our test systems are developed by tailoring enrichment broths to different stages of pathogen cell recovery so we can maximise the chances of establishing healthy, well-grown cultures that produce sufficient antigen for assessment in our immunoassays. Solus kits are routinely used globally in over 30 countries and predominantly in ISO 17025 accredited labs. As you can see on this slide, this gives you an overview of our current portfolio, and if I go through the different kits. Just explain a little bit about each one. So Solus Salmonella ELISA, it's a two-step enrichment, time to result 36 hours. AOAC ISO 16140 certification. Solus Listeria, again a two-step enrichment, time to result 44 hours, and again AOAC, PTM and ISO 16140 certified. Moving on to our newest addition to the range is Solus Listeria monocytogenes. This is again a two-step enrichment and time to result 51 hours, and as I previously said, AOAC PTM certified. We then have Solus E. coli 0157. This is a one-step enrichment, much faster and less manipulation steps than the ISO method, and we currently have ISO 16140 certification here. We then have our two kits, Solus 1 kits, that are, as the name suggests, 1 day to result. And these are our new technology and have time to result that mimic a molecular method. Solus 1 Salmonella has ISO 16140 certification and AOAC OMA, and its extension to scope for confectionery and herbs and spices for challenging matrices. We then have Solus 1 Listeria AOAC PTM certification for environmental surfaces. So a wide range of products that fit most laboratory needs. So I wanted to delve a little bit deeper into the new technology I mentioned in the previous slide. The immunoassay technology that we use in our assays is hugely different to those kits created over 10 years ago. Every aspect of these assays have been inspected, enhanced and developed to optimise its specificity and sensitivity. We as a company recognise that at the heart of every immunoassay are the antibodies. If these aren't performing to the greatest specification, then there's no hope of creating a top quality test. We've therefore took the decision to adopt the most superior antibodies that we could find for each of our assays. Having chose the best antibodies, we developed new techniques to coat our 96 well plates in the most efficient manner, minimising our antibody requirement and simultaneously reducing the amount of non-specific binding to ultimately give much clearer results. The other half of the ELISA sandwich is the antibody enzyme conjugate. Also got a complete overhaul with superior antibodies, optimised conjugation processing and enhanced purification techniques. The resulting conjugate now provides much cleaner, more target-specific tagging, along with the development of a high-quality media. As already mentioned, the resulting assays are far more robust, sensitive, and most importantly, reliable. So these developments have been used in the latest offering from PerkinElmer, the Solus Listeria monocytogenes assay. The assay uses microtiter plates with wells coated with highly specific Listeria monocytogenes antibodies. Any L-mono antigen present in the sample will bind to the antibodies and therefore the plate. The AOC PTM validation shows the assay allows users to employ a single protocol for both food and environmental samples. A simple two-step enrichment process leveraging supplemented media to enhance recovery of the target organism and incubation parameters are shared across all matrices. The PTM also validated the use of the DS2, providing flexibility by offering the use of choice of processing samples manually or with automation. Even if a lab chooses to process the samples manually, the Solus method enables labs to process samples quickly, efficiently and reliably. ensuring a production facility can take action such as release product, reduce inventory, or take prompt corrective action when necessary. And here's a slide that shows the Solus versus the BAM workflow, which shows that results are available up to 2 days faster when using the Solus L Mono. But the real power of our system is when it's coupled with automation. The pathogen detection system, when you introduce a Dynex DS2 unit, it's a fully automated, so you load your samples and reagents and start the protocol. You can just walk away and let the machine carry on the whole process for you. It will load your samples into the plate, it'll incubate, it washes, it'll add your reagents, it reads your results. It's even possible to link the DS2 to the lab's LIMS system. So results can automatically be uploaded and assigned to the correct samples. It's easy to install and train. It's fully set up with all preloaded protocols. It's a real plug and play system. And the real beauty of this system, you can run 1 sample comfortably or 600 samples in 8 hours. And 1 technician can run up to 3 machines. We know that with so many food testing solutions available on the market, it can be difficult to decide which method will be the best fit for your testing needs. But one thing is certain, with our continued technology advancements with Solus kits and media performance, our technology, coupled with an automated platform, makes our system a compelling solution for any laboratory. So please join my colleague Robert Halstead, who will be discussing pathogen detection in today's market at the Meat and Poultry Symposium on October the 6th. And you can find out more information on the Solus pathogen system from PerkinElmer. I'd like to thank you for the time, and please don't hesitate to contact me if you'd like any further information. All of my contact details are on this slide. Once again, thank you.